Okay, well, thanks everyone for joining. I am delighted to be able to introduce to you Dr. Sherry Winkler from the National Cancer Institute, the National Institute of Health. I know Cherie and I have been working together for a few years on the APOL1 gene and its relationship to preeclampsia. I've talked about this many times. She got a bachelor's degree in biology from the State University of New York, followed by a master's in genetics and a PhD in immunogenetics at the University of Maryland. She has spent most of her career at NCI doing a lot of really great genetic epidemiology work in a number of traits, not just cancer. Like I say, more specifically for me, APOL1 and preeclampsia. She's also worked in kidney disease, which that gene is also associated with. She's done a lot of work with HIV and several cancers. So I'll leave it there and I'll hand over to you.
Thank you very much. It's really a pleasure to be at the University of Tennessee, even though it's virtual. I had always hoped that I could visit Memphis. It is Memphis, right? The
Yes.
University of Tennessee, yes. But here I am talking, virtually, and let me see if I can navigate my slides here. Oh, how do I make them go? Oh, I see, okay. And so I have no disclosures. So what I'm going to talk today is about the burden of chronic kidney disease and end-stage kidney disease in the US. I'm going to talk a little bit about APOL1 and its selective pathogen, the pathogen that selected that trypanosomiasis or trypanosomes, a little bit about the APOL1 discovery, and quite a bit about associations with a variety of different renal phenotypes in cohorts that have renal insufficiency, in the general population, in pediatric populations, and the effect of APOL1 in pregnancy. And maybe a little bit about work in progress if I have time at the end. And that's very optimistic because, as you might have heard, I'm actually going to retire in a few months. So approximately 30 million people, or 10% of the US population, have chronic kidney disease, which is defined by a loss of kidney function and measured by estimated glomerular filtration rate, or by renal injury, which is measured by the presence of protein or albuminuria in the urine. So the risk factors for CKD is diabetes, hypertension, heart disease, obesity, and a family history. And I didn't put it on this slide. So being African-American is a risk factor. 680,000 Americans have irreversible kidney failure or end-stage kidney disease. And of these, 475,000 are on dialysis at least 3 times a week. So you'll see that a very small portion of Americans have actually enjoyed having a kidney transplant. CKD is the major risk factor for cardiovascular disease, and even small amounts of albuminuria or protein in the urine are predictive of CVD. Blacks are 3 to 4 times more likely than whites or Hispanics to develop end-stage kidney disease. So I'm just going to pose a question. What would you rather have, ESKD or cancer? So chronic kidney disease leading to kidney failure is an unrecognized health burden. The NIH spends about 10-fold more on cancer research than on kidney disease research, $6,000 million versus $600 million. But within 5 years of commencement of kidney failure, only 42% of Americans remain alive. And the adjusted rate for breast and prostate cancer combined is 50% less than for dialysis patients. So this is a very serious disease. And I have to admit that when I entered this field, somewhat through the back door, I was just stunned by these types of numbers, and including the fact that we spend $20 billion a year on dialysis and treatment for CKD. So the incidence of end-stage kidney disease, and this is in per 100,000 cases per year, is highest, much higher for African Americans compared to any other ethnicity, Amerindians, Asians, or whites. So the African Americans represent 12% of the US population, but 30% of those on dialysis. And the lifetime risk of developing kidney failure is 8% for African Americans. For other ethnicities, it's 2%. And African Americans are a fourfold greater risk of developing ESKD. So if we look at particular causes of kidney failure, the African American to European American incidence ratio is highest for all causes, but it's particularly high for diabetes, for hypertension attributed ESKD, and for glomerular disease, particularly two forms that I'm going to talk quite a bit about. One is called focal segmental glomerular sclerosis, which means that a portion, well, I'll explain this more later, but I'm going to call it FSGS throughout the talk, where the risk is nearly fivefold higher, and for HIV-associated nephropathy. And I'll be calling that HIVAN. HIV-associated nephropathy is really interesting because it is almost never, in fact, to my knowledge, it has never been identified in a person of European ancestry. It's exclusively in Black individuals with HIV infection, untreated HIV infection. And back before the era of HOT treatment, 1 in 10 African Americans would develop a very progressive lethal form of kidney disease. So we hypothesized that the African American predilection for progressive kidney disease, HIVAN and FSGS, was due to genetic variants more frequent on African inherited chromosomes. And at the same time that I was doing this study with HIVAN and FSGS, I was also working with the FIND consortium, looking at a case control study, using a similar approach for diabetic and non-diabetic ESKD. So we elected to use an admixture approach. And for those of you who aren't familiar with this, basically in our African American population, their genomes are a mosaic of African segments of chromosomes derived from the African continent and European derived segments. So on average, African Americans have about 15% European ancestry and about 85% African ancestry. And through subsequent generations, the chromosomes have formed a mosaic of European, African, etc. haplotypes. And the hypothesis, the assumption underlying admixture is that for a disease for which there aren't environmental factors known to cause a disparity between continental groups, Asians versus Europeans or African Americans versus Europeans, that the genetic variants would be more frequent on the population that is more at risk. So in this case, the genetic variants that are causing the susceptibility to HIVAN and FSGS would be more frequent on African inherited chromosomes. So what you would see if you were doing this is that there would be more African haplotypes, African origin haplotypes in the region harboring the disease locus. And you would get a peak of African ancestry in cases, but not in controls. So we did two studies. One, as I just told you, with FSGS and HIVAN, we only had 251 cases and a couple hundred controls. And another study looking at non-diabetic ESKD and diabetic separately. And what we found was that there was this big peak of African ancestry on chromosome 22. And this was compared to either the whole genome or compared to the controls of that locus. So admixture is very nice because you can do a case-only study and compare every point along the chromosomes to the rest of the chromosome as your control group. or you can compare to the controls. And these LOD scores were quite high. We looked at even markers, we looked at odd markers, and we found these really high markers. As I recall, there were about 20 genes under this peak. So collaborators at Harvard, led by Martin Pollack, used pre-release data from the 1,000 Genome Project and identified 3 coding variants that were only found in Africans under this peak, admixture peak, encoding APOL1, apolipoprotein L1. And the only known function for this protein at the time was to confer resistance to trypanosomes. And I'll tell you more about that later. But the variants comprised 2 alleles, which were in absolute linkage disequilibrium. That means they always occurred on different chromosomes. 2 missense variants and 1 6-base pair deletion, which removed 2 amino acids. And these occurred in the SRA binding domain. So, as I said, the only known function of APOL1 was to protect against African trypanosome infection, but 2 species are able to cause African sleeping sickness, or HAT, human African trypanosomiasis. TB gambiense is the cause of chronic HAT, which is endemic to West Africa. And TB rhodensiense is the cause of acute HAT, and it's endemic to East Africa. Actually, gambiense is also endemic to Central Africa. And they protect themselves against the lytic effects of APOL1 by 2 different mechanisms. So these variants restored the ability of APOL1 in human serum to lyse TB rhodensiense, but not TB gambiense. And that's because these variants occurred in the serum resistance associated protein domain that APOL1 was able to bind to. So this is encoded by the trypanosome. It binds to APOL1 and it then activates it. And these 2 variants restored the ability of, or prevented the protein, the protective protein, from binding to it. So APOL1 is in a toolbox of innate immunity genes. And if we look across primates, we see that APOL1 arose in all the human primates, but it's been either deleted in the case of the chimpanzee, our closest relative, or pseudonized. And it's been, so only human, gorilla, and baboon retain a functional APOL1 gene, in spite of the fact that this protein, which arose in all of the higher African primates, confers really excellent protection against trypanosomes. It is highly, highly lytic. And so the fact that, these proteins have been pseudonized or deleted from so many organisms or so many primates suggests that the APOL1s are really toxic, and particularly APOL1. Also, I'd like you to note that the mouse does not encode a ortholog for APOL1, and that makes, in fact, no non-human or non-African primate has ever evolved the APOL1 gene. So there are no animal models to study naturally occurring APOL1. And this has been really made it very difficult to determine what the function of this gene is in humans and why it causes renal injury. So also, I just want you to see that the human, the gorilla, and the baboon are all able to lyse TB brucei, which is a cause of non-human African trypanosomiasis and infects cattle. So the allele structure of the APOL1 variants is interesting, and it's very common. So the GG, the 2G1 variants occur on one chromosome, and because of the close proximity, there hasn't been recombination, and the deletion occurs in another. So these are really additive when you calculate the population frequency. So about 35% of chromosomes amongst African Americans carry, two risk alleles, and approximately 50% carry at least one. So 5 million people, African Americans, carry high risk alleles with a lifetime risk of developing CKD of about 20%. So APOL1 risk alleles are limited to African origin chromosomes. They were carried to the New World by the African slave trade, 1500 to 1900, between 1500 and 1900, by the African diaspora. And now they're being carried by migration throughout the New World and Europe. So if we look at the distribution, we can see them really quite prevalent in African American populations, in Caribbean populations, and in America. And also in South America, but they're particularly prevalent in West Africa. So this posed a real puzzle to us. Why was APOL1 lytic against TB rhodesiense, which is found in East Africa, but it was most common, these variants were most common in West Africa? So we think that APOL1 is under balancing selection in West Africa, and likely by TB gambiense. And I'll show you this in a second, why this might be so. If you carry 0 risk advantage, you have a homozygous disadvantage in that you're susceptible to HAT. If you carry 2 risk alleles, you also are at a disadvantage because you're susceptible to chronic kidney disease, whereas individuals who carry 1 risk allele are resistant to HAT and they don't suffer from chronic kidney disease. So we think that there was a selective sweep on APOL1 G1, particularly in G2 alleles in West Africa. This is a study we did showing these long haplotypes in the Yoruba. And the APOL1 is very frequent. APOL1 variants are very frequent in the Yoruba compared to some of the other populations, Bantu from Kenya or Bantu from South Africa. You can see that the haplotypes are relatively short. And Genovese and Martin Pollock published in Science showing these long haplotype stretches for G1 and G2 compared to alleles in the rest of the genome. So why do we see that G1 is selected for, strongly selected for in West Africa? Well, it's really interesting, the different types of protection and susceptibility that these variants confer. So with the wild type, homozygous for wild types is susceptible to HAT, protected against, or not protected, but had no greater risk for CKD. Whereas if you carry G1, you're susceptible to acute HAT, but you're resistant to gambiense. So gambiense is very prevalent in West Africa, and that might be why these variants are so hard there. You get infected with the trypanosome, but you don't develop disease. You have a latent infection. On the other hand, the G2 variant protects against acute HAT infection by lysing the trypanosome, but it may cause a slight increased risk for gambiense. If we look at the frequencies within Africa, and this is a study that we published quite a few years ago now, we can see that in West Africa, the alleles are very prevalent. The red line just represents 2 copies of a risk allele. Compared to Central Africa, North Africa, East Africa, or Southern Africa. But it's really interesting, in Southern Africa, we do find in certain populations, particularly a population along the South Africa, Mozambique border, where the prevalence rates are approached 25%. So this is really fine grained. Some populations, particularly in West Africa, have very high populations, but others have very low. If we look at the burden of kidney disease in Africa, there are many things that affect it, including epidemiological transition, the high burden of infectious diseases, and as I'll show you, genetic predisposition, primarily from sickle cell trait and APOL1. And if we look, we see that the prevalence in Western Africa is almost 20% of the population have CKD. And this is where the APOL1 allele, particularly along the coast, is highest. So I'd like to just give you a primer about APOL1 so you know a little bit about it without going through a lot of data. APOL1 circulates as a minor component of high-density lipid 3 particles, which is incorporated into trypanosome lytic factor that circulates in everybody's plasma. It's the non-secreted APOL1 is the kidney culprit and the cause of podocyte damage. And I'll show you a podocyte in a minute. Gene expression is driven by interferons. The variants are largely recessive. The high-risk genotypes comprise 2 alleles in any combination, G1, G1 or G1, G2. And terminology that I'll try to use throughout this talk is that low-risk status or genotype is carriage of no or 1 risk allele, and high-risk status is carrying 2 risk alleles. We know that the second hit is required for APOL1 kidney disease to manifest. Viral infections that upregulate interferons are likely second hits. Others might include obesity, large body mass, prematurity, or reduced nephron number in premature infants. So this is just to show you that we can diagnose kidney disease by proteinuria greater than 30, or an estimated glomerular filtration rate less than 60. And the reason that proteins escape is that there is damage done to one of the 3 protein barriers in the glomerulus. And these represent the slit diaphragms, which act as a filter for proteins that get damaged. And this is the purple are the foot processes that you see here of these beautiful podocyte cells that look like octopuses or octopi that encircle the capillaries within the glomerulus. And it's these cells that are damaged by APOL1. So this is just to show you a normal foot process in EM, and foot effacement, which is the loss of these foot processes and the loss of the ability to filter protein. So we did an autopsy series of individuals experiencing sudden death, and we observed a decline in glomerular number with advancing age and those with APOL1 high-risk genotypes, suggesting that the podocyte density on the capillaries is less in people with APOL1 as they advance in age. A mouse model with transgene G0 and G2 experienced loss of podocytes with aging. The mouse model another mouse model also observed podocyte injury and detachment, and it supports a hypothesis that podocyte depletion from the glomeruli is a cause of FSGS and HIVAN. So there's a range, a spectrum of kidney diseases associated with APOL1. The most common and the least severe is global glomerular sclerosis, which you find with hypertension, primarily in African Americans. FSGS, which is scarring of a segment of the glomerulus, and it's focal because only some glomeruli are affected. And HIVAN, which is associated with this proliferation of podocytes and another type of epithelial cell, and the whole thing just collapses. And this is very progressive. HIVAN has odds ratios of 29 in the US, which is really high for a common variant, and it's 89 in South Africa in one study. So the penetrance seems to differ we don't know if it's because of the HIV viral strain or different environmental factors or genetic background. So here we're looking at the percentage of individuals who carried 2 risk alleles compared to background. So we had hypernormal HIV controls who had been infected for 8 years with HIV and no treatment, compared to individuals with high RAN, and fully 72% carried 2 risk alleles compared to a background of 8%, because most people probably moved from this group to that group. For FSGS, it was also 72%, but the odds ratio was lower because the frequency in the population of high-risk genotypes was 13. For hypertensive ESKD, the odds ratio was 7. So these are very high odds ratios for common diseases. Over and over again, we see that individuals who carry 2 risk alleles progress much faster to end-stage kidney disease. This was true in our NIH FSGS cohort, and it was also true in the African-American study of kidney disease and hypertension, where individuals with 2 risk alleles really progressed very fast to ESKD. And sadly, no treatment, nothing could stop this progression. This was a controlled randomized clinical trial to normal blood pressure levels or very low blood pressure levels, and there was no difference. And there was no difference by the type of therapy. So is there utility in screening for APOL1, and when does APOL1 decline in kidney function begin? So we looked at healthy young adults enrolled in the Cardia trial, and we found that the trajectories of EGFR were pretty similar between low risk and high risk groups until they developed proteinuria, and there was a deflection downwards. So albuminuria appeared in the 30s in healthy adults. APOL1 status was associated with earlier onset of albuminuria, and it was associated with a higher risk. And it was coincident with the EGFR decline. So this suggests to me that if we wanted to screen people, it might be best to measure protein in young adults at their annual visits. And then if they have proteinuria, perhaps, especially if there's a treatment available, test them for APOL1. We saw a similar effect with APOL1, with the chronic renal insufficiency cohort, where individuals with a high risk genotype had a, let me see, this is the onset of proteinuria, had a higher risk of developing proteinuria. So proteinuria is a harbinger of kidney function decline. There's no difference in the loss of kidney filtration before incident proteinuria between low risk and high risk, in that top line. There's no difference here. But after the onset of proteinuria, there's also no difference in the rate between individuals who are low risk and high risk. So it seems that APOL1 is damaging the kidney glomerulus, causing protein, which is followed by a loss of kidney function. So this suggests that many factors that lead to proteinuria beyond APOL1 may additionally drive CKD progression. So as I said before, screening would identify those at risk and for EGF decline and possible treatment intervention. So this is the ASK study again, and it just shows again that individuals with standard or intensive goal of blood pressure treatment have no difference in their decline in EGF over time. Okay, so we wanted to know what HIV or what APOL1 is doing in Africa, and I have a collaboration with Bill Wester, who's at Vanderbilt, and they organized a study of about 2,500 participants who were HIV positive, all under treatment between the ages of about 18 and 70. And we did APOL1 genotype. We measured their albuminuria and their estimated glomerular filtration rate. And what we found was that only 6.2% of the individuals, we were expecting 25% to carried high-risk alleles, but 37% of those individuals in the cohort had albuminuria, and 3% had, this is a typo, had macroalbuminuria. This is nephrotic range. That's indicative of HIVAN. So when we looked by ethnicity of the APOL1 genotype, we found that it was in the Hausa Fulani who were living in this particular region of northern Nigeria, that only 2.1% carried high-risk genotypes, whereas the Igbo and the Yoruba, it was 49% for Igbo and 14.5% for Yoruba. So this shows that even in one geographic location, the frequency of these variants vary greatly by ethnicity. And we also found that APOL1 high-risk genotypes had an odds ratio of about 2 for microalbuminuria, about 4 for macroalbuminuria, and it was also strongly associated with chronic kidney disease measured by EGFR. But it didn't account for all of the microalbuminuria because, you know, so few people carried it, only 6% carried it, but 37% had microalbuminuria. And that was really a surprise to us. So now we're looking more closely. And this is also a randomized clinical trial. So the individuals who do have proteinuria, and this was measured at 2 time points, are going to be put on ACE inhibitors to see if we can resolve proteinuria and maybe slow progression to kidney disease. And I probably don't have to tell you that if you develop chronic kidney disease in Africa, it's virtually a death sentence. There's very little in the way of dialysis and very little in the way of kidney transplants. So the doctors there are amazing at keeping people alive without treatment for you know, but yeah, you know, it's very sad for me to go there and realize that some of these countries have 2 nephrologists for a whole country, that people, there's no treatment available. Okay. Okay, so I probably told you before that there's a second hit required, but particularly for collapsing forms of FSGS, if you give individuals interferon for treatment for example, HCV infection, individuals who carry true risk alleles are likely to develop a form of collapsing FSGS, which is very similar to what we see with HIVAN. And so at the beginning of the COVID epidemic, I thought to myself, gee, I wonder what's going to happen with COVID and APOL1 carriers. And indeed, there have been, I hope I have a slide for this. I don't. So the COVID-19 pandemic, there have been 11 cases to date of people with African ancestry who carry true risk alleles who have developed a form of collapsing glomerulopathy that is reminiscent of HIVAN. And in fact, it's now been named COVAN. And what I'm wondering now is whether individuals who don't develop HIVAN will be like individuals with chronic HIV infection and perhaps develop renal injury that will, you know, cause them to develop CKD later in life. So what about APOL1 and kidney transplantation? Well, it's all in the kidney. Okay. So if you take a high-risk donor, the kidney in the recipient will have decreased allograft survival, as you can see in this graph, that compared to low risk donors, I believe the odds ratio, here it is about 2. The hazard ratio is about 2 for developing kidney failure in the recipient of the allograft high-risk kidney. On the other hand, if the donor is low risk and the recipient is high risk, there's no effect on the survival of the kidney. So what this suggests, one actually makes 2 points. One is that the form of APOL1 that is the culprit that's causing the renal injury is probably intracellular, and that circulating APOL1 has no effect. And secondly, that we have to take a deep look of what the consequences would be of screening donors for the APOL1 genotype. And because kidneys are so short in supply, and even a bad kidney, might survive for 3 or 4 or 5 years, it might be worth giving those kidneys to someone who's older or more dire straits. On the other hand, by telling people that they can't give kidneys, or no, let me rephrase that. Right now, there is a point score for how you evaluate kidneys and whether they can be donated and who they can be donated to, where you give poorer quality kidneys to older people and better quality people to younger people. And part of that score is ethnicity. If you could type people for APOL1, it might mean that there are more kidneys that are available for transplant because you're taking out that ethnic across the board descriptor of kidney allograft survival potential. And so what about living donors? So if you have a high-risk genotype, does that mean that you might be more susceptible to developing hypertension or chronic kidney disease or kidney failure? So in a collaboration with Lonnie Ross at the University of Chicago, she went back through the registries and looked for donors who are now on the list for the registry to get a kidney. And we found that 55% of individuals who need a kidney and who are donors carried 2 risk alleles, compared to 22% of healthy donors and 13% of the general population. And so we don't really know what the denominator is for this, and we don't know what the absolute risk is, but it does suggest that there may be a liability of being a living donor if you have a high-risk genotype. Okay, so there was a second study that we did with Mona Doshi. And in this study, we looked at 136 Black living kidney donors who had a median time since donation of 12 years. And these individuals were approximately the same age by risk status, by genotype, about the same number of men to women, the same body mass, systolic pressure. The only thing they differed on was EGF. And even though this is robust, less than 60 is considered CKD, they were still significantly lower for their EGF compared to low-risk donors. So this is pre-donation and post-donation status. If we look at low-risk individuals, they had a higher EGF before donation compared to high risk. And after donation, they had a smaller rebound for the high-risk group compared to the low-risk group. So I've already told you this. So amongst the low risk versus high risk group, kidney failure was 11% in the high risk group compared to 0% in the low risk group. And CKD was 36% in the low risk group compared to 58% in the high risk group. And EGFR was much lower. and they were more likely to have proteinuria. So does kidney donation increase the rate of EGFR decline? So here we can see from baseline that it appears that high-risk donors had a greater decrease in EGFR. And if you take that out, by the time they get into their 40s or 60s, they might have ESKD. However, if we look at high-risk APOL1 genotype in non-donors versus donors, we see that the lines are parallel. So they're not losing faster, they just did not have the same rebound and they started off lower. So for APOL1 and living donors, it's probably ethically imperative to quantify the risks of kidney donation to APOL1 living donors and to quantify the risks of recipients of high-risk kidneys. To address this, the NIDDK has funded a longitudinal study called the APOLLO to enroll every living recipient, donor-recipient pair in the US, as well as deceased donor-recipient pairs over a 2-year follow-up period, a 2-year period, and then follow them up and see how their kidneys perform over time. And I think this is going to answer a lot of questions about the long-term consequence and give us some absolute versus relative numbers. I mean, even though it seems relatively that having a high-risk kidney is not good, the absolute risk might be quite low, and we just don't know that yet. So APOL1 is also associated with pediatric kidney disease. This is a study from the Neptune cohort of children with kidney disease, particularly nephrotic or glomerular kidney disease. We had 199 African Americans, and we looked at FSGS in those with high risk versus low risk. And we found that only 25% of kids with nephrotic syndrome had FSGS in the low risk group compared to 89% in the high risk group. And FSGS is one of the more severe forms of kidney disease in kids. We also saw other associations. So children with high risk genotypes were more likely to be obese, and they were more likely to be premature. These might act as second hits. The age of onset for APOL1 associated FSGS was 12 to 14 versus early childhood, younger than, generally 8. For the other glomerular diseases, they were more likely to have hypertension, and they were more likely to have left ventricular hypertrophy and cardiovascular disease, 53% versus 12%. They had a steeper decline in EGFR and more proteinuria. And these studies have now been replicated. So the epidemiological relevance of the risk genotypes in African Americans, the population attributable risk is the fraction of disease that would be eliminated if the factor weren't present. And the explained variance is the proportion of disease explained by genetic factors among all the risk variants that contribute to the disease. And I compared this to smoking for men, where the attributable fraction is 90% for males and the explained variance is 12%. And here we can see that for the variance for HIRAN is 37%, the attributable risk is 68%. For FSGS, it's 18 versus 68. For hypertension, it's 7 and 52 for attributable risk. And if you look at these numbers for the lifetime risk of carriage of 2 risk alleles, it's 1 in 2 for HIRAN, 1 in 24 for FSGS, and 1 in 9 for hypertensive attributed to ESKD. So this has considerable, APOL1 is placing a considerable burden on the African American community. So there are several, excuse me, let me get a drink. There are many proposed disease mechanisms. The one thing we know for sure is that interferon upregulates APOL1, and when APOL1 is upregulated, cells die. in animal models and in cell models. So one is that there's an opening of the plasma membrane of cation channels. And this is similar to what we see with lysis of trypanosomes with APOL1, where it inserts itself into the membrane and causes lysis of the cell by opening up channels. Inflammasome activation resulting in cell death. We've seen that in animal models. Endoplasmic reticulum stress from incorrect APOL1 folding. We also see that in vitro cell systems. Protein kinase are activating global suppression of protein synthesis. We published a paper on that a few years ago with Jeffrey Kopp. Mitochondrial dysfunction that's been observed, and reduced ubiquitin levels and therefore higher levels of APOL1 have also been observed. So it's very difficult to know what the true mechanism is here, or maybe all of these are functional. Animal models have not been that helpful. Cell models, it's overexpression, they haven't been that helpful. My personal feeling that it's probably a combination of 3 and 5, that there's ER stress from incorrect folding and severe mitochondrial dysfunction, which results in a stressed podocyte. Okay, so we've long been interested in other phenotypes, and we were motivated to look at APOL1 associations with preeclampsia. Our first study that we did, and I'm not sure that we published this, was about 10 years ago, where we looked at moms with preeclampsia, and we didn't see any association with APOL1. But the motivation for the study was that preeclampsia, which is a defect in the placenta that leads to hypertension in the last term of pregnancy, and it contributes greatly to mortality and morbidity of mothers, really globally. So it's more severe and prevalent in African Americans. It accounts for 20% of pregnancy-related deaths. APOL1 is highly expressed in the placenta. There are higher circulating levels of APOL1 in preeclamptic moms, and there are anti-APOL1 autoantibodies in preeclamptic mothers. So for infants, 15% of infants born to preeclamptic mother are premature. 30% have intrauterine growth retardation or restriction. And both of these, small size and prematurity, result in a lower endowment of nephrons, which might predict CKD at older age in these children. Also, APOL1 transgenic mice, both G2 and G0, developed a preeclampsia form when the transgene was connected to a promoter that expressed in the placenta. And it was actually the fetal genotype that drove this phenotype. So we did 2 studies. We had 121 infant pairs from New York City, and we found an odds ratio of 1.84 for fetal genotype and a non-significant association with maternal genotype. We reached out to Bob Davis and Claire Simpson at University of Tennessee, who were involved with the candle, and asked if we could look at their cohort. And this started a very nice collaboration. And we were able to replicate this association in a very nicely controlled study. Again, the odds ratio was about 1.92 for the fetus, and for maternal carriage of high-risk genotype, it was less than, it was non-significant. So I think that Bob Davis calculated that 1 out of 8 preeclamptic births in African Americans is probably due to APOL1 high-risk status. So this was ongoing work, but it's no longer. We wanted to look for interactions between maternal country of origin and maternal fetal APOL1 genotypes in another cohort, the Boston birth cohort. So we had 426 Black mother fetus pairs and 213 pairs that were matched on age, parity, and fetal sex. So the analysis comprised of genotyping them and comparing APOL1 fetal high risk versus fetal low risk. And we also looked at mother fetal APOL1 genotype mismatch versus a match. So what I mean by that is if the fetus was carried a variant and the mother didn't, or vice versa, the mother carried a variant that the fetus didn't, that would be a mismatch in both directions unilaterally. And what we found that there was amongst the African Americans an increased risk of preeclampsia for the babies with an odds ratio of about 3.2. And there was also an additive association of about 1.7. There was an interaction with country of origin. We also saw increased preeclamptic risk with a mismatch of 2.7. So if there was a mismatch, they were at increased risk independently of whether or not the fetus carried the APOL1 high risk genotype. Now, when we looked at Haitians, we saw no effect of the fetal genotype on preeclampsia incidence. This is also true if we looked at country of origin. Many of these were individuals were born in the continent of Africa, not just the Caribbean. And we also saw no effect of APOL1 fetal maternal mismatch on preeclampsia incidence in the Haitian group. So we really don't know what this means. It was kind of, to our minds, a shocking finding. We looked at what we could look at for this particular study. We looked at the principal components of population substructure of ancestry, and we saw no clear difference between our Haitian group and our African American group. We weren't in this study able to look at socioeconomic or demographic factors, and we also didn't look at the role of micronutrients or overall maternal health. But we plan to do that, and in the process of writing for funding to do such a study. We also don't know what the penetrance of APOL1 is on the African continent. And you have to think to yourself that a risk factor that causes preeclampsia and loss of fetal life or maternal life is going to have a big reproductive fitness cost and be selected against, by purifying selection. So, when you look across Africa, and I haven't really shown you a fine-grained map, but I guess I did show you some of the frequencies. I keep thinking to myself that maybe there's something special about certain populations in West Africa that allowed them to tolerate this very toxic protein and these toxic variants, whereas the genetic background in other parts of Africa are less tolerant. Maybe the penetrance is going to be less with APOL1. And there's some data now coming out of Africa suggesting that the penetrance of APOL1 on kidney disease is less than it is in the United States. These studies, for the most part, are cross-sectional rather than longitudinal. And we know that the association of APOL1 with CKD in a general population is pretty low, that really it's a progressive factor. And it's also possible that we have ascertainment bias, that people who are sickest don't show up for these cross-sectional studies. But the data that's to date, the data that seems to be coming out is that it's less penetrant. And if that's due to environmental factors or G times G interactions, G times Z, we don't really know at this point. My bet is that it's going to be a little bit of a combination of both, that epidemiological transition in Africa, obesity, diabetes is going to potentiate the penetrance of APOL1. But there are probably also, as yet undetected genetic factors that are influencing the effect sizes. So just to close up, I've shown you that APOL1 is associated with, quite strong odds ratios across the life expectancy. It's associated with prenatal CKD in children with HIV, with lupus nephritis, transplant survival, arterial-nephrosclerosis, and hypertension attributed CKD. Surprisingly, in the older adult population, APOL1 does not seem to be such a high risk. And that's possibly because the people who are most susceptible have already developed disease in their younger life. But it'd be very interesting to look at APOL1 across the lifespan in some of these long, big cohorts, longitudinal cohorts that have been assembled. Okay, in the interest of time, I won't go through all of this, except to say that for genetic screening, there should be clinical trials to see what the utility is for screening for APOL1, testing for proteinuria, studying in young adulthood, and clinical trials with ACE inhibitors and RAS blockers to see if individuals with proteinuria might be able to reverse their proteinuria and delay the onset of EGFR decline in CKD. So I won't go into future work except to say, let me see now, this one. You probably didn't notice when I showed you this before, but the blue are the cases and the green are the controls. And the percent of African ancestry is highest in the controls and lowest in the cases. In other words, the cases have more European ancestry compared to controls. So one of my hypotheses is that the variants on European haplotypes potentiate APOL1 penetrance, or alternatively, African haplotypes attenuate APOL1 penetrance. So we're now doing a extreme phenotype study design to see if we can identify some of these modifying genetic factors if they indeed exist. So I have a number of collaborators. I won't go through all of them, but this is my lab group a couple of years ago. It's a little smaller now. And these are my collaborators, including Claire Simpson and Bob Davis here at University of Tennessee, who have been so really helpful and wonderful to work with on preeclampsia. Bill Wester, who I did the Nigeria study with, and Jeffrey Kopp, who got me into this field in the beginning. So thank you very much.
All right, that was great. Do we have questions? So I will ask a quick question to get people started, because this is probably obvious and you probably get this all the time, but so do humans need APOL1?
Oh, I'm glad you asked me. I am glad you asked me that because I forgot to mention it. There is one human who lives in India who developed trypanosomiasis from a TB evensi pathogen. He should have been resistant to it. So because of that, he came to the attention of the medical community, and they looked, and he had a knockout mutation for both alleles. He was homozygous for a knockout. He expressed no APOL1. He had no renal phenotype, no phenotype of anything. Humans do not need APOL1. So that's an N of one, but an important N of one. He's in his 50s now, a very, energetic, healthy gentleman.
So I guess then that's the, is that a strategy to try and reduce the levels of APOL1? I guess you have to do it pretty early in life. And how hard is it to target an apolipoprotein?
Okay, so there is a silencing RNA being developed by a company. I don't recall the name of the company, but they made transgenic mice expressing APOL1. They were able to get high rates of proteinuria at the levels of nephrotic syndrome. And then they introduced the silencing RNA, which is able to reverse the proteinuria, and the mice do not develop kidney disease. And if they take that away, it continues. So, I think there's going to be a treatment available, but whether it's going to be available to the world or not, I don't know. If you think about it, in Africa, trypanosomes are endemic. And if you knock out their APOL1, you might knock out their defenses against a really lethal pathogen. So, I think that maybe other strategies might be better and maybe cheaper. One of the things I didn't say is that the lifetime risk is 20%, but that means 80% who carry these high-risk alleles are never going to develop kidney disease over their life expectancy. And so personally, what I think APOL1 is doing is it's damaging podocytes and stressing them. And then when you get CKD or other stress around the kidney for whatever reason, you progress faster, you develop more renal injury. HIRAN and FSGS are subsets of that, where you have an onset of nephrotic range proteinuria and a rapidly progressing condition. For those, I can see using silencing RNA to reverse the course and just stop it. We don't know what the inciting factor is, but for arteriolnephrosclerosis and progressive kidney disease associated with non-diabetic kidney forms of kidney disease, maybe renal protective drugs like the RAS blockers or ACE inhibitors would be fine, and lifestyle modification. I mean, there have been no clinical trials to really look at this, unfortunately.
Thank you for that.
Any other questions?
Sherry, do you want to talk just for a second? What do you think of the role of HLA in potentially modulating the impact of a fetal-maternal mismatch?
Oh, yes. One of the reviewers, when they reviewed our paper, brought that up. And I absolutely think that HLA might play a role, and that if we were going to look at the role of APOL1 and HLA, I don't know that they would be synergistic or epistatic, but I do think that they would be potentially additive, the roles of HLA. And I also like the idea of looking at, and I think I've talked to you about this before, that with kidney transplantation, and you can, at fetuses, an immunologically protected transplant in some ways, and differs, by a number of paternal alleles that from the mom, that it might be global mismatch. It might be the number of surface expressed cells, alleles that differ between the mother and the fetus that also contribute to preeclampsia. And I think that we should look at that with the GWAS, with our GWAS data eventually.
Great.
I don't know, what do you think, Claire?
Claire, you're on mute.
Sorry. Sorry, what was the question?
Oh, the question is, whether HLA might interact with APOL1 to potentiate preeclampsia. And then I started, just talking about global mismatch across the genome
Right.
of surface expressed alleles that aren't shared by the mom.
Yeah, it's a tricky question. I have a policy in my life, which is to stay away from HLA at all costs because it's so complicated. But I think it's an interesting question. I think it's definitely something that needs thought about and studied.
Yeah.
But I think we need a lot more data on the different segregating HLA haplotypes as well. I don't think we've gotten a lot more data, but I still think we need more granularity than we have.
The person really to talk to is Mary Carrington. And she's a friend of mine, and she's also probably the world's expert on HLA and has done studies on preeclampsia. That is her world. She's at NCI, and we might want to involve her if that's a question that we really wanted to dig into.
Okay, that sounds really
because I agree with you. HLA is so complex.
Yeah, I kind of feel like you have to use people who have that specialty. You just end up just blundering in and making a mess otherwise.
No, I mean, you can do a GWAS and see a lot of hits on chromosome 6.
Yes, but it's like...
to dig deeper, you really need an expert on board. So, it's a suggestion. If you were going to write a grant to look at HLA, I would strongly recommend that you approach Mary Carrington.
Okay, cool.
She is
right.
really
Put that on my list of things to do.
Well, I might be having dinner with her tonight. I could mention it to her.
Oh, please do.
Okay.
I'd love to speak to her.
Okay.
Nice to see you again, Sherry.
Nice to see you too. Bye. Thank you so much.
Thank you very much.
Yeah, well, thanks. If there's no more questions, I'm sure everyone's eager to get into their holiday weekend.
Bye.
Okay. All right.
Yeah,
Bye-bye.
thank you so much. Thank you for your
Okay.
time. It was fascinating
Bye-bye.
work and talk. Yeah.